Everyone, it's Mr. Sinti, and today I'd like to explain to you the movement of phloem sap throughout trees or plants in general. And you know what's interesting right out of the gate about phloem sap is that I can't say that this particular discussion is about the ascent of xylem sap, because xylem, which mainly conducts water, the water flow direction is generally in an upward direction, if you recall, because it's being pulled by transpiration, so it's the ascent of water up a plant. Whereas when we talk about the water that contains a lot of sugar and organic products of photosynthesis, we can't say that it's ascending or descending. What we can say is the movement within the phloem of phloem sap is translocation. So that's what that is. Because what's fascinating about it is that, like for example here, you can see in this picture, let's just jump right into the discussion. I'm so excited, I'm getting ahead of myself. And so phloem, like just take a look at this picture. This is a cross-section. You may recognize it. It's a cross-section here, this part at least is, of a stem. And you're like, what kind of stem? Is it monocot or dicot? Well, the vascular bundles are around the perimeter, so this is clearly a dicotyledon. And this cambium layer right in here is where the cell layer right here, which is meristematic. The cells are capable of division. They'll either become xylem or they'll become phloem right there. And so what's interesting is that if you look at this, I mentioned that water generally flows in an upward direction like this. So the water is moving up. Like for example, you can see here, the xylem is coming up like that. It's coming up like that in the plant. But the phloem, it's like, well, what direction is it? It's showing, the arrow is showing that it's coming down like this. And so it could be. But the question is, where's the sugar being produced? And so what we'd like to talk about is what's the source. And I know what you're thinking, you know, isn't that obvious? The source is usually the leaf, is where organic molecules are produced during photosynthesis, like glyceraldehyde phosphate, and then glucose, and then ultimately sucrose is the main sugar that's being transported within the phloem sap through translocation. But basically it's from source to what we call sink, and the sink could be a fruit or it could even be the root. But depending on the season, the movement of sugar could be going in an upward direction or a downward direction. So we call it just simply translocation, if you're okay with that. And so the phloem movement is a process called translocation. So let's get into that conversation. And so first off, a little review of what phloem is. It's composed of two main cell types. One of them is called a sieve tube or a sieve tube member. And what's interesting about it is, though it's alive, it's a hollow kind of cell, and so it's a good conduit for carrying phloem sap. But like, wait a minute, if it's hollow, how can it survive? Well, it partners up with this little cell next to it, adjacent to it, called a companion cell. And so it's able to shuttle material through plasmodesmata to keep it going. And so in angiosperms, flowering plants that produce seeds and a fruit, the phloem functions in translocation, and the sieve tube members are the ones that are conducting the phloem sap. And as you can see, they're going through the sieve tube plates right here. This is a cool scanning electron microscope photograph of that, showing the sugar moving in between cells. This is transmission electron micrograph. Notice here how the sieve tube cell is mostly hollow, but yet there's a connection here by the plasmodesmata, and so the cytoplasm, any energy that it needs, any protein that it needs, is provided by the companion cell. Isn't that nice? It's like having your own little assistant or secretary, the companion cell. How nice. And so phloem sap is aqueous. So I don't know if you're thinking that this is like powdered sugar, granulated sugar moving here. Now it's very, very sweet, sort of like Kool-Aid. And it's an aqueous, meaning it's dissolved in water. And the primary sugar I mentioned is the disaccharide sucrose. And so it's not just sucrose. There could be some hormones that move along here. Plants produce hormones, chemical messengers, even some amino acids. So here's a great example of a sugar sink, like a fruit. We all know, for example, that the fruit contains a lot of sugar, but fruit doesn't make the sugar. So check this out. So the sunlight comes down like that, and it strikes the leaf. And in the mesophyll cells, in other words, the middle cells, the palisade and spongy mesophyll, you get those parenchyma cells that are producing sugar, and then through photosynthesis, and then that's being conducted through the vein. And inside the vein, excuse me, inside the vein is the phloem. And so this is what we're talking about, translocation of phloem sap. And like, where's it going? It's going here. And so this is considered, the fruit is considered to be the sink. In other words, that's where things are being dumped into. So it's the consumer of the sugar or the storer of the sugar. That's what the sink is defined as. And so phloem translocates sap from the source, which is often a leaf, to a sugar sink, which is generally either a fruit or a root. But as I mentioned at the top of this discussion, in contrast to the unidirection, in other words, one direction of xylem sap, which is from the root up, phloem sap is somewhat variable. And so what we can say, though, is it goes from its source to the sink. So it's where it's being drawn from and where it's going. And so generally the source is the leaf. Generally the sink is the root or the fruit. And so how does it all happen? This is a great picture. It shows that sugar is being produced here in the mesophyll cells of a leaf, and then they get moved over into the phloem, which is vascular tissue in the vein. And then the phloem just simply moves down like this, moves down the petiole, and then ultimately it moves down the stem, and then it goes and it gets deposited in the sink, whether it be a fruit or a root. And the mechanism, I'll look at the detail of this in a moment, but the mechanism is that water from the xylem comes over and sort of pushes it down that way. So it's under a positive pressure. That's what's moving phloem sap. A translocation is positive pressure. It's kind of cool, like in a maple tree, you can tap it. Again, you don't have to stick this tap in very far. You're like, well, why is that? It's a big tree. It's got to get inside it. Well, inside the center of the tree, if you recall, is the heartwood, which is xylem, and then the sapwood, which is xylem. The phloem is really close to the perimeter. It's just below the bark. Remember, bark is made up of cork, the cork cambium, and then the phloem. And so you just have to go just a little bit, like an inch, into the tree, and next thing you know, it'll start dripping out phloem sap into the bucket. So it's amazing. You have to do this once in your life. This occurs all the time in Vermont. And so this sort of Kool-Aid is a little too much water. And so what you then do with these buckets is that you heat them and evaporate the water, and it becomes very sugary. And that's how you make maple syrup. You make maple syrup using phloem sap. There you go. So every time you're putting syrup on your pancakes or your French toast, think of phloem sap and think of this video. or not, no worries. So, here, here's a picture of a tuber, which is a modified stem. We like to simply call it an onion bulb. And so I may have misspoke. I'm getting goofy here. Tuber is, for example, a potato. This is a bulb right here, but it could be the sink. In other words, like the onion could be photosynthesizing, producing sugar, and it gets stored here in the bulb. But then, in the second year, this is a biennial plant. In the second growing season, that actually becomes the source to grow the roots and the shoots and the flower. So it really depends on the season whether or not the bulb is going to be a source or a sink. So just to sort of mention that. And so the sugar source or sink usually receives sugar in the nearest location. And so this, I find this to be a detail, but kind of interesting, that even on the upper leaves of a branch, those leaves could be sending the sugar in an upward direction to the shoot tip, which needs sugar in order to grow. That's where the primary apical meristem is, the terminal bud. But then just a little bit lower in the branch, the lower leaves in the same branch could be taking the sugar and having it go down into the root. And so even within the same vascular bundle, some sieve tube members can have phloem going in an up direction and some can be going in a down. So it's kind of wild. Translocation. It's everywhere, everywhere. It's going in all directions. And so sieve tubes can be going in one direction, the opposite one could be going, or the other one could be going in the opposite direction. And so, the conversation ultimately is going to come down to this, which is how can the sugar get uploaded from mesophyll cells into the sieve tubes? And you're like, well, doesn't it just move over there? Yeah, it does just move over there, but the sieve tube members are really, really sugary. They can even have like 2 to 3 times higher sugar content than the mesophyll cells in the leaf that are producing it. And so it's going to require some effort to do that. You're going to be having to push sugar into the sieve tube members, and that requires energy. So it's active transport. Active transport is how sugar is uploaded into phloem. And so how does that occur? Well, it occurs using a proton generated gradient which drives this. And so I'm going to, if you don't mind, I'm going to come over here if I can, and let's see if I can. I'm going to call up this screen right here and sort of illustrate this. And so here is a mesophyll cell. So this is a cell in the leaf that's producing, in the chloroplast, this is the one that's producing sugar. So I'm going to go here, sugar. And I'm going to abbreviate the sugar. I'm not going to go through the whole, though I can. I'm not going to go over into the Calvin cycle and say that that's glyceraldehyde phosphate, and then twice around 6 carbon dioxides produce glucose, and then and then it ultimately produces sucrose. Okay, so I'm going to go S for sucrose. And so over here is phloem. All right, here's a phloem cell, so a sieve tube. So what I'm saying is that there's a lot of sugar already present in the phloem, like this. And so how's it going to work? And so what the phloem does, it's really, really neat, worthy of our discussion. What the phloem cell can do is, check this out. It could use a proton pump. And let me just go over what that means. A proton pump is taking protons like this and pushing them over to the other side like this. And so now you're creating over here, over in the mesophyll cell, a gradient. So it's becoming real positive over here and real negative over here. You're creating a membrane potential. You're creating an electrochemical gradient by doing that. You're pushing it, pushing it, pushing it. Now, this is the active transport. You're pushing it, and that requires energy to do that, ATP. So you're pushing it over. Now, why in the world do you do that? Because ultimately over here, you got this cool membrane protein. And what is happening here is that this particular protein over here is a co-transporter. And so check this out. A sucrose could fit in here like this, and then these protons are itching to get back in because they're in high concentration over here. These protons are in high concentration. So they can't get back, can't get back. So what they do is they go over here, and what happens is this is how the phloem is able to receive a massive amount of sugar, and it can keep pushing it even though it's very, very concentrated. So the cell expends energy to create a membrane electrochemical gradient. Then it uses that to shuttle sucrose over into the phloem. Now, as that in and of itself doesn't move the phloem, the sugar solution. What actually does that is, now that you've created this really strong hypertonic environment, do you remember, like adjacent, let's go over here, adjacent to the phloem is the xylem. And the xylem's got lots of water, and the water's going like this. And so the water says, hey, look at that very hypertonic environment over there. Maybe I'll go over and investigate that through osmosis. And so water movement occurs. And so the water is then flowing into the phloem, and that's causing pressure, which then drives the phloem sap away from the source. And so here's the sieve tube plate right here. And so then this is what is pushing. This is positive pressure, which is pushing the phloem sap down. Hopefully that's clear. I find that that's a really cool phenomenon like here. And check this out. A little video clip illustrating this maybe. And so here you can see the xylem, which is conducting the water is ascending in the xylem, and it's being pulled by transpiration. So that's a negative pressure, pull. Negative pressure is pulling it up. Cohesion, adhesion, capillary action. All right. So then look, check this out in the sink. So the mesophyll cells of the leaf are producing sucrose. And okay, so then they're being pulled into the phloem using active transport, because you've created this little gradient right there. And then the water on top of it pushes, do you see here? Now this is positive pressure. It's sort of pushing the sugar in a downward direction like this. Check that out. And so the sugar water is moving. This is the process of translocation. You're watching this animation, translocation. And now what's going to happen is it goes from source to sink. And so at this point, the sugar is being offloaded. In other words, you sort of arrived at your station and you're getting off the train. And so notice how the phloem down below here is becoming less hypertonic, and so the water then flows back. So you're creating like this little circuit here. So very sweet up here, not as sweet down here because it's being collected over in the sink over here. And so look how fast it's going, especially in the daytime. So that's kind of interesting. So just wanted to point that out. Let's come back over here if we can and finish this conversation that we're having. So what I want to say about that is this proton gradient helps to move it, and this is a picture from a textbook with regard to this. I'm not sure how clear it is, but basically we're talking about how the cells expend energy in the form of ATP in order to create an electrochemical gradient. This is a proton pump, and it makes it very positive over here, and so and negative inside. And so when the hydrogen ions, which are protons, get attracted in, it's a co-transporter which then pulls the sucrose across, even though it's highly concentrated in the sieve tube. And so as a result, what you see here is the source, the sucrose is being offloaded into the sieve tube. The water is pushing it down. So phloem sap moves by positive pressure. The high levels of sucrose in the source lowers the water potential, and so there's a higher water potential in the adjacent xylem, so the water then moves over, and then the removal of the sugar down below creates an increase in water potential, so therefore the water moves back into the xylem. And so downstream of the sieve tube is where the sugar is offloaded, like for example in this beet, and then water just moves through osmosis always. And so I hope you enjoyed that conversation about phloem sap, and maybe after watching this you want to celebrate with a stack of pancakes and some good Vermont maple syrup. Thanks for watching.